we're going to move on in the risk assessment paradigm to consider TTC. The development of the TTC dataset was one of the great undertakings within the Cosmos program, and there are two main elements to it which you'll hear. The first is the development of an enriched dataset, and you've heard the basis of that. That's why, one of the reasons why we went out to collect data, and it's gone into the Cosmos database. And the second is how we can apply this data, which are on the whole oral data, through to data that are or exposure scenarios, but for the dermal route. I think what we're going to focus on here primarily is the development of the TTC database and some of the quality control processes that went through to that. And the next speaker is Chihae Yang, who will describe that.
The Cosmos non-cancer TTC database and its approaches to cosmetics and extension to the cosmetics chemical space. And because I'm not sure your background, how much you know about the TTC, so I actually still put in quite a bit of background information, and I'm sure a lot of you are so sick and tired of it because I've been talking a lot of times on this. So please bear with me if you have heard me before. The TTC, although this might be the first time you hear, or not very many times you heard it before, this is a very well-known concept of 5 centuries of dose make the poison, starting from this famous person. So this is back in the 15th century. Now, in 20th century, 1995, US FDA fifth, and if you actually walk into that OFAS building, in their first floor, they have this huge wall-size TOR displayed, the threshold of regulation. What it tells us is that they could think about a exposure concentration so low, exposure to the humans so low that they may not expect to see appreciable risk to human health. Okay? So if that was set at 0.5 ppb, and this is the regulation, and so you could actually opt to take this route when you have a component in your formulation. If it's below this exposure, you could go ahead and file for it. The only caveat is that you have to be publicly open because that's what TOR is. Since then, FDA and with the others, the fragrance industry and the EFSA and all the others, they're trying to expand this TOR concept to bring some structure information and more rationale. So in 2004, Crows and others, it's the first time they actually combined the cancer part, what we call cancer TTC, started from Mitch Cheeseman when he was at FDA back in 1999. And then this is the part that Monroe, back in 1996, proposed. And this was the combination. TTC, I'm going to concentrate on this one on a non-cancer part. And it actually consists of 2 parts, very important 2 parts. One is chemical space. So for those of you who are chemists, should be happy. There is a chemistry here. Then there is a toxicity data part. So it's a bio domain and the chemistry domain meeting. The structure categories of these chemical structures, that was actually proposed back in it was actually 1979 something by Kramer. So I'll go that later on. The structure class is class 1, 2, 3, and you all know this by Kramer classes. Safe, innocuous, or most could be related to unsafe. So we categorize structures into 3 parts. Then we look at the tox data, and we come up with a very pragmatic decision tree. So this first chemistry part, it will start out as this a decision tree. Does this chemical have N-nitrosol, triazone, or quad? Okay, and there's a split. It comes down, it asks 30 some questions. And you all probably are very familiar with the tox tree, and it's asking the series of questions, and it was 1978, published by Kramer and others. And at that time, he already has seen, for Kramer classes 1 structures, going through this, Noel was already much higher than Kramer class 3. So he was actually, at that time, it was actually only 80 compounds. Kramer and his colleagues established this rule using only 80 compounds, was being able to separate them. Now, in 2000, I'm just quoting Sue, this is one of many, many publications out there. Kramer class 1, 2, 3, and is pushing the cumulative distribution, okay, toward more potent. So like I said, TTC is a exposure-driven method that actually knows chemistry. It has to have some chemical information. So the chemical space has to be well defined. So then that goes back to where? It's a database. The database was first established by Monroe in 1996 paper, and how many cosmetic chemicals were there? There were about 190 of total 613. It's not bad at all. Monroe is a wonderful, amazing set, really. I don't know how he got that lucky, but he got just about everything in there. Very diverse set. Not too bad, but we wanted to expand more to actually represent cosmetics chemicals more, and if we put more related to cosmetics ingredients and the formulations, will that threshold change? That's a reasonable question to ask. Next one is, as we all know, I told you we eat lipsticks. So cosmetics are actually applied dermally, but all our testing results are oral, and there's a reason because the oral toxicity is more, two reasons actually. One is more reliable than a dermal toxicity, and another is just the bottom line. So these are the ones, the Karen Blackburn from they have come out with a small set. They looked at it and they thought, oh, this is actually good chance that it'll work, but we need more database. And Andrew Wolf actually looked at our first preliminary data set, and he thought that, yeah, this is promising. And about this oral to dermal, there's a close approach. We're going to be concentrating on the first prong, the expert group 1. We actually established expert groups. Again, this was very instrumental to have our scientific advisory board from, I'm still talking about Susan Felter, who actually recommended to us that we need to have 2 separate groups for this. So expert group 1 takes care of the database and the framework, and expert group 2 took care of this oral to dermal exposure, the extrapolation. And this is a very, you should be by now, you should recognize right away, this must be one of those crazy, I was looking for all data, right? Here's what it is. This is, you already saw that, oral tox data. So we have all the different data sources. Then it went to the oral repeat tox. And why is everybody's asking about this data quality? Why is inclusion criteria are important? Because you know now why people are asking. NTP has their protocol. FDA has their protocol, right? SEC have their protocol. We don't even use same uncertainty factor. ECHA has their protocol. Everybody has their protocol. Different. And I'm here. I'm a beggar. I don't have any data, so I have to grab all I have. So what do I need? I have to understand each of their criteria and to make sure that find the common things so that we could put them together in one pot. That's what happened. This is why this was difficult. Then, from this, we took Noah's out first, the candidates, no observed adverse effect level. Before we use it as the point of departure, we first get that part out. And this is not TTC database yet. This is just a candidate. Okay? Then into this Noel that we pulled out of this toxicity data, we add Monroe, and as you know, EPA IRIS has all their RFDs, the oral reference dose. Okay? And that's also based on NOELs or BMDLs. So it goes through assessment. We add those data, and then the EPA ToxFDB has some of those data are actually their OPPD, the data evaluation records from the submission. And so those data are valuable, so we have put that together. Then we're not done yet, because I'm still just trying to grab as much as possible. And we went through this filter, but that's a data record filter. That is not actually interpretation of the results filter. That comes in here. So we formed a group, remember this expert group one we formed? They then said how to take Noel values. Then after we have pulled them out, some of those, they actually review them. So this is that review session. And we have 9 cycles of data going back and forth, and there was 4 formal QC sessions. So I am just summarizing this again. When we talk about the toxicity study inclusion criteria versus the Noel criteria, they are slightly different. Study inclusion criteria, this we already talked about it while we talked about in the overall repeat dose toxicity. Cosmos study inclusion criteria have been applied. So we have actually many toxicity data included. How many animals we should test, what's the duration of that, and on, what the study parameters are. These are records. And this is easily able to control. Okay, so that's that. Then comes the Noel selection criteria. It gets getting harder. At that point, we have something called reliability criteria. When we talk about reliability, things get here a little more subjective, isn't it? Because it depends on your perspective, whether you're at EPA looking for things or whether you are SCCS looking for things. Your perspective actually changes. The reliability criteria is slightly different. And in this one, so we always have, okay, what is our reviewers' clinician score? What is their opinion? This was very important. Then we go, there are 2 ways again to it. Remember that when I talked about the old repeat dose toxicity, what was the most sensitive experiment? Subchronic rat. There are people still who would go for subchronic rat when they do Noel values out. Okay? They go for the low L, and then they found the corresponding Noels. We could have done it that way. We tried to be using chronic because Monroe's first approach was chronic, and if it's chronic study, then we don't have to apply any uncertainty factor of not going from subchronic to the chronic type of what we call adjustment. Because anytime we make an adjustment, there is a question mark. So that's what we decided to do as a group. Now, in that case, still we go for the lowest Noel that has a clear Noel and Noel have. Okay. So remember, I said there's 2 ways to do it. One is go for the Noel first, subchronic Noel, the lowest Noel, and get the Noel. That's one group does that. Another one we did it was go for the chronic, and we went for the lowest Noel that has clear Noel. Clear Noel means that if the Noel is the highest dose tested, then you have a question mark because you don't know whether you're really there or not. Maybe you're already all above, or whether you're not in the right range. Okay. So these are the things we have to be considering, whether it's a freestanding or not. So that's all written in. These are the things that you could actually quote it and pull the numbers out, and that's what I mean by we could do that candidate. But that's not quality yet. The quality comes in here. This is the statement that is so lovely statement, and it makes our life so difficult that if we are not a toxicologist. Toxicologically meaningful and human relevant, and the mechanism is known. This is our charter. So in order to define toxicologically meaningful, we had to learn a lot, but then we had a group of experts who tell us what to do about it. And we could not QC whole 500, 700 studies. That's not possible. So what we did was we defined what the section that we must actually look at. Okay? It turns out to be about 25%, and I'll go to the next table. This is very boring, really. Okay, but it's important for us to go through this so that you appreciate when you actually get this data set. Because this data set will answer, in fact, what Catherine was asking. Here I have a liver. What is the Noel distribution? Here I have a endocrine. What is the Noel distribution? Which one actually gives you a lower Noel? I could tell you endocrine gives a lower than liver in our database. For right now, how to answer those questions? We have to have a quality database first, right? So QC1 started out, the first time I started out actually more than 700 candidates in the beginning. Then we applied this rules and then goes down. But the first thing we actually looked for was the lowest 10%, the most potent one, because that's going to drive the threshold. and the ones that the variability is high across all the databases. And at this point, we didn't even look at kernel class 1, 2, 3 because we know at that time the data were just all, we're looking at the whole thing. We were not ready to actually carve the 1 to 3 yet. At that point, we had 140. That was our version 1.6. So it actually came down from 700 something to 1.6, 460 compounds real quickly. Right at that time, we realized that we need more cosmetics ingredients in it. So this is when Mark and I actually formed a group and, harvesting group, we need to do this thing. Okay, then what we have done, all that, after we harvest the new chemicals, new cosmetics-related ones, then we took this over again. Now this time by the Kramer classes 1, 2, 3. The 10% most potent Kramer class 1, 10% Kramer class 2, 10% Kramer class 3, most potent, and the ones in each group, the high variability. From if TOXFDB and Monroe gave 100 times different NOEL, we had to look at it. If TAPA and SCCS have 1,000 times, of course we had to look at it. Those things happen. So going through that, we looked at 57. This means 57 compounds. This means that we have looked at least 150 studies. It's not read, actually looked at. People read and write notes. Some people write dissertation length of opinions, trust me. At least they publication length. Then while this is going through, what we also find out was there was a great idea, which is, well, why don't we actually combine Monroe and cosmetics together, Cosmos together, and so that we could actually expand our whole database in using one, right? That's a very reasonable thing to do. And so we want to first make sure that does it really affect, how much does it affect Monroe when we add more cosmetics in it, okay? It sounds so good, but you have to think about this. When you add something to something, which one, and if there's an overlap, which one of your kids are you going to keep? That's a Swift's choice, remember the movie? Very difficult choice. That's why we had to do QCs all over again. Here we go. That went on, but then it got smaller because we did much of that that we were Cosmos people were able to do because we had a toxicologist, and these were the actually ones that we could not resolve, and we asked the experts. And the more important thing, there are chemical classes that are very, very difficult called the parabens and phthalates and other things. This one's one, we really needed the group to write a strong statement for this for us to move on. Because we've been going at it for more than a year on this. So this last one, actually what happened was Cosmetics Europe came in and helped us out because that's when the group met at Cosmetics Europe's office and they went through, they realized that they really needed to make a decision and that was good. So those were made. So right now, I am pretty sure this is my final because the time is running out. If you give another 10 years, we'll still be cleaning, so we should not do that. 1.9, this is the final one, is 556 compounds. In terms of, there were another slide that some of you saw with the 1.8 values, and the values don't change very much in between because we are now at a stable stage, and the TTC thresholding method itself, the fifth percentile itself, is quite rigorous. I mean, it is actually, you have to pull all the fifth percentile out before it actually see a huge difference. That's stable they are. I mean, they are stable. So this is good. Of those 556, the cosmetics inventory, there are 495 is in the cosmetic inventory, and I told you 190 was already in Monroe. So Monroe is amazing. Back in 1996, he already had that insight. Kramer class 1, 2, 3. There's a slight difference here. This is a nice difference. We have more class 1 than Monroe does. In terms of class 2, this is really not enough. 38. Monroe had 28 of them. We had at least 38, but it has compounds that are quite difficult to handle. So I'll show you. Nutrients and the lipid-soluble vitamins and essential amino acids, we decide to remove them because biologically this is not how how the regulators usually regulate. They don't use TTC for those. There's a human data and there's another mechanism going on. So usually these are not really relevant, so our group decided to remove it. Monroe, on the other hand, still kept it because they're not actually, he was a more general database. So when we put Monroe back in, will it go in? I have to actually ask one last time, our Aegis expert group again. Will I put it in? Probably will not, okay? So these are see how little this thing takes time, so you have to think about it. Now, the compound categories we actually struggled with and, looked at it. Hair dyes, because some of the hair dyes just hit the genetic tox alert so well. But then it doesn't mean that they are genetic tox. It's just that it hit them. So because of that, we were arguing, debating back and forth with that. We actually looked at the analysis with and without and found that there's no effect, really. This is how stable the thresholds are. So we decided to just put them all in. That's no problem. 110. Monroe had 13. Parabens and phthalates. We have quite different values than Monroe, but which is very understandable because Monroe was published back in 1996, and there was a period that endocrine disruptors and these things were not really, we were not really honing in there yet. Okay, so we thought tox was the problem. Really, all this problem toxicologists give us, okay, right? But that is not so. We thought the chemistry was always good, so simple, right? Now, like I said, when we started adding things, Monroe had his Kramer class 1 to 3. He's an expert. We had Kramer class 1 to 3 by tox 3, right? So when I add in, you think that it'll just fit right in, it would have been a really beautiful case, but it wasn't. Monroe's Kramer class 1 and tox 3 Kramer class 1 wasn't the same. They were like 40-some compounds, quite different. So we had to resolve that. But resolving it, meaning we have to understand what the reasons were. So the errors, the reasons, but one of the things that I want to actually focus on and have your awareness is that when you use this type of software, there's a certain compound classes you should be aware. If it is one of those, just maybe we shouldn't try software. I'm a software person, so trust me when I say it very carefully. Some of the soft forms is a problem, sodium and potassium and all this, because that actually, sodium benzoate and benzoic acid are two different classes. Does it make sense to you? Okay, we have to consider that. Okay, sucrose alkyl esters. They are very hydrolyzable. For the people in a food area, understand that this is hydrolyzing it. So they should not be, when they hydrolyze, it should not be 3. But when they're looking at the whole thing, it's going to be at a 3. That's the problem. These things we have to be careful. And then there's the tautomeric dyes. So none of the people are good at it. Tox 3 is not good at it, or Monroe wasn't good at it. And because we actually have Stevie Enoch and Kirk Robinson and I, 3 of us actually did this analysis. So we know these things we have to be careful of. When you get this 3 types in cosmetics. So it will be different if you're in a fragrance. There will be different types, and it's already published. In cosmetics, when you do this type of thing, hair dyes, tautomeric dyes, you want to be careful. Okay, again, my favorite, look at things about, this is our TTC data set, and after 9th cycle, I don't think it's going to change much anymore. I'm so happy about that one. I don't have to make new slides. So here's my categories, okay, and here's Cosmos, who's in Monroe. You can see some of the organohalides. Monroe has more. You see, Monroe is a general industrial chemicals. We don't put or eat organohalides. Here we go. The phosphorus we don't have. US, no stirring. That's good. But we have some surfactant cations. And another decision we decided is that we would put organosilicons in this database because we know how to handle it, and we know the toxicity. There are some good toxicity data. So usually, TTC does not handle organosilicons. And if you go to food, they don't put it in. But in this database, we do have them. So once you have this set, one thing we do is, again, the uncertainty factors. So this is why we have to be careful with this word uncertainty because it means so many different things. When you say uncertainty from RFD, they go, well, 10 for the intraspecies, 10 for the interspecies, and 10 for the, so the subchronic to the chronic. Everybody uses different kind of things, so that becomes 1,000, right? When you go to SCCS, sometimes they like to use lowest NOEL, factor of 3. Sometimes they like to use subchronic to chronic, sometimes they don't. So these are the things. This becomes now manipulated. Manipulated is not a bad word. This is a calculated NOEL. So we will divide it by subchronic to chronic data. This is what Monroe did. So if it's given during the subchronic day time for 3 months, okay, at 90 milligram per kilogram per body weight per day, then we will actually divide it by 3 and then it becomes 30. So now you would remember this. Subchronic rat was the most sensitive species, and you take that and divide it by 3, it becomes really sensitive. Okay, and then 2 times, so there's a total 6 for the 28 days, 6 times. And then we decided, we did quite a bit of experiments on this reproductive developmental on the Monroe and Cosmos, and Subala was our leading person for that. She actually is a reproductive toxicologist, and we decided it doesn't make any difference, so it's just cleaner not to apply any adjustment. And then this is a low-L to no-L, because if there's a good low-L, we don't want to lose that data, so this is what we're doing. Some of you already seen this, and then the reason that I don't put a number on is because you will see the number on our publication. I would like to have our experts to see that number first before I give a talk, so that's why. But these numbers are final. So what I'm saying, the number is actually the median numbers and the 50th percentile. So I'll actually go over the pattern with you. So we added about 360 new substances. So there was 190 in Monroe. Now we added about 360. It's a good size now. So if we combine Monroe and Cosmos together, we have total 960 substances. It's a pretty good size database. It's actually a large database. It takes so much time to read everything to do a QC. Okay, then for cream class 1, 2, 3, many of the Monroe substances are also related to cosmetics, which means that a lot of the cream class 190 was actually Monroe 1, quite a few, okay? So there are a lot of overlap. So remember, this is what I'm saying, this is a Sophie's choice, which one am I going to take? The Monroe overlap substances were reevaluated. We had to evaluate because we cannot just guess, right, to decide which one. And then we did all this 10th percentile evaluation, and we found that is actually makes more difference. The quality and the relevance of that NOEL decisions, the POD decisions with the critical of that makes more difference than any adjustment factors. Reproductive developmental adjustment didn't make any difference. This one, okay. So I just want to know that you remember this. The Monroe threshold, class 1 is 3. The class 1 is the least worrisome one, so that should be higher. And then class 3 is the most, let's just say, unsafe, but this is quote unquote, 0.15, right? And in between this, Monroe class 2 was 0.91, but remember Monroe class 2 was only 27 or 28. This class 2 are just not reliable numbers. Now, what happened to the Cosmos? If I just look at the Cosmos data set alone, class 1 is higher than Monroe. So verbally, I could give you a number, it's 3.33. So don't quote me, please. I want my expert group to see that first. Then class 3 is higher than Monroe, much higher than this. I'm very happy. It's about 10 times higher than that. Okay, it should be. It's a cosmetics ingredients that we eat. Okay. One interesting thing was actually this class 2. Class 2 was actually lower. 38 compounds and 27. First of all, the distribution is not very reliable. Another thing, there are 2 compounds that were actually that low. All the others were higher. Guess what those 2 are? They are allyl heptanoate. Oh, boy, I hate that compound. I'm telling you. This one was like better than I don't know how many times we evaluated and on and on. Very difficult. And then there's this compound. Those are the only 2 on this side, very stubbornly. We just left it there. And this were QC 3 times. So you just went through with me, like, what did Steve say? Mark Millen's PhD in 10 minutes. My 5 years of misery in how many? Okay. This was very interesting life, actually. And then, this is the exciting part. The exciting part is when the things are done, then you get to use in a very, very easy way. So here we are. Here's the TTC data set. It's going to export the TTC data set, either Cosmos or Monroe or together, but it's joined. So we placed one or the other, which our group decided. Then there is a one TTC unit. So you put it on. So I'll show you real quickly on that one. Here's the current status, and this one is just take-home message. But I just rewrote the same thing over again so that at the end, the Cosmos TTC, whatever it's taking. This part is this oral to dermal exposure part, the extrapolation. There's a separate group doing that, and there's still some work needs to be done and still going on. And we also, another one that I like to tell you is that putting it all together, we are actually working with Matt Dent on the level 3 case study with quercetin, because it has the kinetics, the pathway dependent risk assessment, and it has the quercetin kinetics of the availability, so that we could compare that using this. All this you'll be able to access it through the Cosmos TTC. This has been built and tested in a recent Cosmos meeting in July at Liverpool, right here actually. And so ever since then, we've been just testing and QCing, and it will be released when Mark said it's released. So this is my trouble compound. If you look at the data, the safety evaluation, if there was any decision by the SCCS or there's a RFD or MOS, it'll show up here. So I'm just going to go toxicity data. There are a lot of data. The point is, there's enough data that gives you a headache. Okay, so I mean, we could do that, or I'm going to come back and to show you benzoic acid. So what do I mean by TTC database? So first of all, the TTC database was built to give you a plethora of the view for safety evaluation. So this actually captures if there's an RFD decision, or there's an MOS decision, or there's an ADI decision. So, like, for example, for this one, there's an POD and NOEL values. The owner is the SCCP, and MOS is 206. And coming down here, there's an RFD. Okay. So this is going actually toward the safety evaluation, high-level decision-making type of information. And this information then is showing up here as safety assessment data, and these are the ones that where we actually collect all the calculated POD and assessed NOEL and LOEL values. Okay. So very easily you could negotiate in and out. One thing I like to show you is too easy. Here, computational workflow. So here we are. What do I mean by expert structures and data? This is like when you want to get NOEL values out, HNL values out, or toxicity data out. This is what you will be able to use it. TTC export. Because I've been promising you this, a very simple one. So here we are, Cosmos, Monroe, or Monroe extended by Cosmos. Okay? So let me just say Cosmos. It's going to take just a little longer because it's not still a performance. We have not optimized our performance. Right now we're concentrating on cleaning up all the content. Okay, so I could say save, or I could say open. And it'll be Excel sheet. your beloved Excel sheet is coming at you with all the Cosmos data. Where did it go? Oh, it must be. Yeah. Oh, you know, I think what it is it's the resolution. So it's I cannot see this. Let me make it small. Because I wanted to want to show them.
Save it on there.
We'll just download them. Yeah, we'll just save it then. Yeah. Okay. Is it doing it? No, it's just, I think it's busy. It would have been nice to show them. What is this?
So it's still, we're still doing something somewhere.
Yeah, so where is it going on? Because usually it'll just download. Click on it. Do you have a download? Say export, it should actually just download it or save as, and it'll be an Excel sheet, and it'll give you all the full, what the compound actually had, what the data, what study it was associated with, critical study, and what is the critical effects, what is the Noel value of that, and what is the calculated Noel value of that, and what is the uncertainty factors it used. Because one of the biggest problems we had with Monroe, we had to guess. So it's giving all those things and the references, and then what it also gives is very important thing, target organs and Noel calculated and original Noel. So this, through this, you could answer Catherine's question about how is liver compared to thyroid in a Noel or Loel profile. So that's one, and then another one that I was going to show you is the TTC workflow. You open it up, and you either put smiles or more file or whatever, and and then your EDI, estimated daily intake. Then you'll go through and say, well, this is a class 1 compound, it is over that, below this. It will actually run through the table, the image that you saw from the cross paper, the decision tree.
Thank you very much, Chihae. We'll see whether we can get I wonder whether it's because it's a temporary account as well and it won't open a
I think what it is
the
WebEx thing that it's just memory-wise.
Yeah.
It's not a huge file. It shouldn't take.
Yeah. Okay, so to summarize, we've done an enormous amount of work on TTC, enriching this data sets, and not only that, but creating a process for the quality control, which in TTC, something which wasn't formally established before, and as has been alluded to, the historic data set was one which was just published with no further data, so we've very much improved on that. Any questions on the TTC approach?
I was just wondering whether perhaps I've missed it somewhere, or whether there are plans to revisit the Kramer classification, because it seems like now you're sat on this amazing data set, but we're still effectively binning chemicals by something from the late 70s, and surely with all the kind of tools and techniques that we've talked about today, it would be of merit to look at that data again, see if we can classify the chemicals in a different way that would give us more refinement in terms of then applying the TTC practically.
Did we actually fix this question? Yeah. Okay, this is a gold question. We have a plan. We had a plan, and this is still actually part of it. One of the deliverable is the potency categories. So when I showed you that my favorite, the classes and the bar chart, right? That is actually, it's not just anything. It is actually that categories. So we first find potent categories. And then the next step is what Steve is doing. What we are trying to do is trying to then put mechanistic understanding into the potency categories. Then eventually that will replace clinical classes. We have to do one step at a time. And until then, we need a working system.
Yeah, and I think to supplement that, there was a large piece of work, which I'm certain you're familiar with, that done under the auspices of RIFM, last year and the last couple of years with Terry Schultz and Dave Roberts, where they manually went through a large number of data, or a large number of structures, and assigned them. And as well, Steve Enoch has had a go at assigning compounds. One of the, idealistically, one of the aims would be to replace Kramer by a mode of action means of classifying compounds. Are we near that? Well, we've got the technologies, we've probably got the database now, but it hasn't been done. And I guess what you're talking about in reality is we have something that, for all its faults, is possibly acceptable or accepted. And if you're talking about replacing it, well, it will be a very long road to push a complete revision or complete replacement of Kramer. So yes, we have the technology, we have the data to do it, but it hasn't yet been achieved. Any other questions in the room on TTC? So we can hear someone.
Yes, hello. This is Amaya Idisa from IFRA. I was just wondering whether you are able to tell us a little bit more about the TTC and extrapolation from oral to dermal exposure, and when that aspect will be available to disseminate.
I will attempt to answer that question. The question, if it wasn't caught, was something more about the oral to dermal extrapolation. Can I go back up? Can I use the slide? I'm just searching for, Chihae did have a slide, I think it was the previous slide to that one. Okay, I'll address that question orally. The second ILSI EU expert group has been attempting to address the oral to dermal extrapolation, and they've done that through a decision tree which leads the user through potential use scenarios. What you may, if you can see the screen, what you may be able to see is on the right hand side of the screen now, it says tiered approach for extrapolating oral TTC to dermal exposure, and that builds on the Crow's approach of understanding possible exposure and leads you through a decision tree, so whether the chemical is in the TTC chemical space, the exposure scenario, compare with oral TTC, predictions of oral bioavailability, comparison with oral TTC, and so forth. My understanding is this approach is being submitted for publication in regulatory toxicology and pharmacology pretty much as we speak in the next couple of weeks. So that will come out fairly rapidly. And as an aside, as part of that, the skin permeability database, which were compiled to support this approach, will be out in the final version 2 of the Cosmos database at the end of the year. So I hope that answers that question or addresses a little bit. If not, please email us and maybe we can talk later on.
Just as a follow-up to that, because early in the morning, I think it was when during the presentation, the initial presentations, you did show the use of the PAMPA approach for determining the oral or predicting the oral GI absorption, and I was wondering whether that could actually be incorporated into this flowchart you're actually showing now on the slide, and therefore incorporate the oral absorption and refine the extrapolation of oral to dermal even further.
Currently it isn't, because we're very conscious of the issues of using PAMPA and a prediction of PAMPA, or even a measurement of PAMPA. So this mainly concentrates on the dermal route of exposure. Areas of investigating PAMPA were really to enable us to identify compounds with both high possible bioavailability following oral exposure and low. So if something had very low permeability from pampa, then we would be able to flag that as compared to maybe something that was strongly absorbed dermally. And to cut a long story short, we're not familiar with any or really compounds that fall into that have high dermal bioavailability as compared to low oral bioavailability. So that's one aspect we did follow on in the early parts of this research.